Japan has a 4% obesity rate. The United States has 42%. That is not a small gap. That is not explained by portion sizes or willpower or the kind of generic dietary advice that fills health content on the internet. That is a tenfold difference between two of the wealthiest, most developed, most food-abundant nations on Earth. Countries with similar access to calories, similar urbanization, similar work schedules, and in many cases, similar stress levels. And here is what makes it scientifically remarkable. It is not genetics. When Japanese people move to the United States and adopt a Western diet and lifestyle, their obesity rates rise within a single generation to match the American population they live among. The protection disappears when the food changes. The biology responds to the environment, not to the ancestry. Japanese Americans eating American food become metabolically American. Japanese people eating Japanese food in Japan maintain the fourth lowest obesity rate on the planet. This means the answer to Japan's metabolic advantage is sitting on their breakfast table every morning. It is in the specific foods they prepare, the specific way they eat them, and the specific cultural habits around meals that have remained largely unchanged, while the rest of the industrialized world abandoned their equivalent food traditions for a processed food system that has produced a global obesity epidemic. But here is the dimension most content about Japanese food ignores entirely. The same population that maintains a 4% obesity rate also has the longest life expectancy on Earth. These two facts are not coincidental. They are mechanistically connected. Visceral fat, the fat that accumulates around internal organs and is almost entirely absent in traditional Japanese populations, is not merely an aesthetic problem. It is a metabolic active tissue that pumps inflammatory cytokines into the bloodstream continuously, driving insulin resistance, cardiovascular disease, type 2 diabetes, and cancer risk simultaneously. Eliminating visceral fat does not just change what someone looks like. It fundamentally changes the biological environment in which every cell in the body operates across decades. The populations with the least visceral fat live the longest. The Japanese have the least visceral fat among all developed nations, and they have the longest life expectancy among all developed nations. The same foods that prevent fat accumulation are the same foods that extend healthy life. Understanding one means understanding both. Today, I am going to break down 7 specific foods at the center of this system, the mechanisms that make each one work, the research behind each claim, and the practical way each food fits into a daily eating pattern that does not require discipline, restriction, or calorie counting. At the end, I am going to show you why the way these foods are eaten together matters as much as the foods themselves, and why replicating individual items without the structural eating pattern around them produces only a fraction of the outcome. Let us start with the biology that most people never hear about. The visceral fat problem, and why it connects directly to longevity. Most people think about body weight as the relevant number. The research on longevity disagrees. The Okinawa Centenarian Study, the most comprehensive longitudinal investigation of extreme longevity ever conducted, tracking over 1,000 centenarians across 40 years, documented something that confused the researchers initially. These individuals were not all lean by Western aesthetic standards, but they had near zero visceral fat. The fat that accumulates around organs, behind the abdominal wall, inside the liver, the fat that no scale measures and no mirror shows, was simply absent in a population that regularly reached 100 years old, with intact cognitive function and cardiovascular profiles that their doctors consistently found remarkable. Visceral fat is biologically active in a way that subcutaneous fat is not. adipocytes, fat cells, in visceral deposits secrete a specific profile of inflammatory cytokines. Tumor necrosis factor alpha, interleukin-6, and resistin. These compounds circulate continuously in the bloodstream, driving the chronic low-grade inflammation that the longevity research identifies as the primary upstream driver of every major age-related disease. They impair insulin signaling in muscle and liver cells, initiating the insulin resistance cascade that precedes type 2 diabetes by years or decades. They damage the endothelial lining of blood vessels, initiating the arterial injury process that eventually produces cardiovascular events. The centenarians of Okinawa were not managing these pathways through medication or structured intervention. The traditional food system that sustained their population had simply never created the conditions for visceral fat to accumulate in the first place. 7 specific foods were at the center of that system, and the mechanisms through which each one operates on fat storage, fat burning, and metabolic health have now been documented in peer-reviewed research. Mechanism 1: gut microbiome remodeling and fat storage suppression. The first mechanism requires understanding something about the gut that most diet culture never addresses. The bacteria living in your intestines are not passive passengers. They are active participants in your metabolism, directly influencing how many calories you absorb, how much of what you eat gets stored as fat, and what hormonal signals reach your brain about hunger and satiety. Research consistently shows that the gut microbiome composition of obese individuals differs systematically from that of lean individuals in one specific way. The ratio of 2 major bacterial families, Firmicutes and Bacteroidetes, is skewed toward Firmicutes in individuals with higher body fat. This matters because Firmicutes bacteria are more efficient at extracting calories from the same food. A gut dominated by Firmicutes extracts more energy from every meal. A gut with a higher Bacteroidetes ratio extracts less, producing a structurally lower caloric uptake from an identical diet, without any change in what or how much is eaten. The traditional Japanese dietary pattern is, arguably, the most powerful gut microbiome optimization system ever identified in a real-world population. Food. 1. Natto, 50 grams daily. Natto is fermented soybeans. Soybeans inoculated with Bacillus subtilis and left to ferment for approximately 24 hours, producing a sticky, stringy food with a strong flavor that most Western palates initially resist and most Japanese have consumed since childhood. It is eaten at breakfast with rice and soy sauce, sometimes with mustard, exactly as it has been for centuries. The research on natto and visceral fat is among the most specific in the entire fermented food literature. A study published in Molecular Nutrition and Food Research demonstrated that natto intake reduced visceral fat in a dose-dependent manner in animal models, meaning the more natto consumed, the greater the visceral fat reduction. The fermented version was substantially more potent than plain soybeans. Fermentation unlocked biological properties that the raw ingredient does not possess. The same soybeans, the same nutrients. The fermentation process changed what they did in the body. The mechanism runs through several simultaneous pathways. First, the Bacillus subtilis bacteria in natto survive stomach acid and reach the intestine alive, unlike most probiotic supplements, which are destroyed by digestive processes before reaching their target. Once in the intestinal environment, these bacteria directly shift the Firmicutes to Bacteroidetes ratio toward the lean metabolic profile. Second, natto suppresses fatty acid synthesis in the liver while promoting fatty acid catabolism, meaning the liver produces less new fat from dietary inputs and breaks down more existing fat simultaneously. Third, natto delivers nattokinase, an enzyme with documented fibrinolytic effects on cardiovascular function, alongside isoflavones that activate estrogen receptor beta in multiple tissues, and spermidine, a polyamine compound documented as one of the most potent dietary inducers of autophagy, the cellular cleaning process that the longevity research identifies as a primary mechanism of biological age maintenance. The Okinawan centenarians, consuming natto several times weekly, were not supplementing spermidine or probiotics. They were eating breakfast. Every single biological mechanism the supplement industry now targets with individual compounds was being delivered simultaneously through a single fermented food that cost less than €1 per serving. Food. 2. Miso, 1 bowl daily, unpasteurized. Miso is fermented soybean paste. Soybeans fermented with koji mold over months or years, depending on the variety, producing a deeply savory paste that dissolves into warm broth to create the foundational soup of traditional Japanese cuisine. The key word when purchasing miso for metabolic purposes is unpasteurized. Pasteurized miso has had its bacterial content destroyed for shelf stability. Unpasteurized miso contains the living lactobacillus bacteria and the enzymatic activity that make it biologically active. A bowl of miso soup delivers simultaneously probiotics from the living bacterial cultures, prebiotic fiber from the soybean substrate that feeds those cultures, isoflavones that support metabolic function, and the soup matrix that slows gastric emptying and contributes to the satiety, signaling that prevents overeating. One preparation, multiple mechanisms, consumed daily. The traditional Japanese meal structure ensures that miso soup arrives at the beginning of the meal, before the larger caloric items. This sequencing matters. The liquid volume, the fiber, and the bacterial signaling compounds all reach the upper digestive tract before the main caloric load, establishing the hormonal environment, lower ghrelin, higher peptide YY, earlier cholecystokinin release, that determines how much will be eaten in the subsequent minutes. Food 3. Fermented pickles, tsukemono, 30 to 50 grams daily. Every traditional Japanese meal includes tsukemono, pickled vegetables fermented in rice bran, salt, or miso. Not vinegar pickled, which kills the bacteria. Traditionally fermented, which preserves and concentrates them. The specific bacteria in nukazuki fermentation, rice bran pickles, include lactobacillus strains that are particularly effective at producing short-chain fatty acids, including butyrate. Butyrate is the compound that the gut microbiome produces from fermented fiber that performs two critical functions simultaneously. It strengthens the intestinal barrier, the tight junction proteins that prevent bacterial lipopolysaccharides from entering systemic circulation and triggering the inflammatory cascade that drives visceral fat expansion. And it signals the brain through the gut-brain axis to reduce appetite, producing genuine satiety rather than the caloric deprivation that conventional dieting requires. Butyrate is not a supplement. It is the byproduct of a gut microbiome being fed the fermented substrates it was designed to process. Subscribe to Longevity Code here. Every week we decode one longevity population, one food system, one set of mechanisms. Always peer-reviewed, always practical, always something specific you can start this week. Mechanism 2. Brown fat activation and fat-burning thermogenesis. Most people have one type of fat. Technically, they have two, but only one is metabolically active in the way that matters for fat burning. White adipose tissue stores energy. Brown adipose tissue burns energy. It contains mitochondria-dense cells that generate heat by uncoupling the proton gradient from ATP production, consuming stored fat as fuel in the process. This is thermogenesis. Infants have large amounts of brown adipose tissue. Adults have very little. But adults retain something that has become one of the most exciting targets in metabolic research: the capacity to stimulate white fat cells to express UCP1, the uncoupling protein that gives brown fat its thermogenic function, effectively converting fat-storing cells into fat-burning cells in a process called browning. Two specific compounds in traditional Japanese foods have been documented to activate this process through distinct mechanisms. Food 4. Matcha, 2 to 3 grams daily. The distinction between matcha and regular green tea is not merely cultural. Standard brewed green tea steeps the leaf in hot water and discards it. You receive the water-soluble compounds that have diffused out during brewing. Matcha is the entire leaf ground to powder and consumed whole. The concentration of EGCG, epigallocatechin gallate, the primary catechin responsible for green tea's metabolic effects, in matcha is dramatically higher than in brewed tea, because you are consuming the entire leaf rather than a water extract of it. Research has documented that EGCG significantly increases the mRNA expression of UCP1, UCP2, PRD-M16, and CPT1-beta in brown adipose tissue, the genes that govern thermogenic function. EGCG activates AMPK in brown adipose tissue, the cellular energy sensor that regulates the thermogenic gene expression cascade. In animal models of diet-induced obesity, EGCG supplementation produced decreased body weight gain, reduced plasma and liver lipids, and higher body temperature alongside elevated mitochondrial density in brown adipose tissue. The preparation method matters more than most people realize. Matcha should be whisked into water heated to approximately 80 degrees Celsius, not boiling. Boiling water denatures the delicate catechin structure, reducing EGCG bioavailability significantly. The traditional Japanese preparation of matcha, whisked carefully in warm water, consumed as a focused ritual rather than a rushed drink, was inadvertently preserving the biological activity of the compound through correct temperature management. Adding a squeeze of lemon to matcha increases EGCG absorption through the vitamin C interaction that stabilizes catechins in the alkaline intestinal environment. Adding dairy milk binds catechins to casein proteins, dramatically reducing absorption. The Japanese do not add milk to matcha. The biology rewards this choice. Food 5. Wakame seaweed seen 5 to 10 grams daily. Wakame is the soft, dark green seaweed that appears in miso soup and seaweed salads throughout Japan. It contains a compound called fucoxanthin, a carotenoid pigment unique to brown seaweeds that has attracted significant research attention for its effects on adipose tissue. Published research demonstrated that fucoxanthin induces the expression of UCP1 in white adipose tissue, the conversion of fat-storing white fat toward fat-burning brown-like activity. This finding was notable because UCP1 is normally expressed almost exclusively in brown adipose tissue, which is minimal in adult humans. Fucoxanthin achieved UCP1 expression in abdominal white adipose tissue, the fat surrounding internal organs that drives visceral fat's inflammatory burden, producing measurable reductions in abdominal fat weight in animal models. The mechanism appears to run through fucoxanthin's accumulation in visceral adipose tissue specifically, where its metabolite, fucoxanthinol, drives the UCP1 expression and also improves insulin resistance by regulating cytokine secretion from abdominal fat cells. The same compound that burns visceral fat also reduces the inflammatory signaling that visceral fat produces. The Okinawan centenarians, consuming seaweed daily at every meal, were not targeting their UCP1 expression. They were adding wakame to miso soup because that was what the recipe required. The abdominal fat that never accumulated in this population reflected decades of daily fucoxanthin delivery from a food that costs almost nothing and appears in every traditional Japanese meal. Mechanism 3. Structural eating patterns and insulin management. The first two mechanisms operate through specific bioactive compounds. The third operates through the behavioral and structural framework that determines when and how those compounds are consumed, and through the carbohydrate choices that determine the insulin environment in which fat storage decisions are made. Food 6. Purple sweet potato as primary carbohydrate. This requires addressing what seems like a paradox. Japan consumes significant carbohydrates. White rice is eaten at nearly every meal, and yet the population maintains metabolic profiles that Western nations cannot approach despite consuming far fewer total carbohydrates. How? Part of the answer is in Okinawa specifically. The traditional Okinawan diet built its caloric foundation not on white rice, but on purple sweet potato, consumed as 70% of total calories in the traditional diet. Purple sweet potato has a glycemic index of 44. White rice has a glycemic index of approximately 72. White bread is over 70. Table sugar is 65. A glycemic index of 44 means that the same caloric load of sweet potato produces a dramatically smaller and slower postprandial glucose spike than the equivalent amount of rice, bread, or refined carbohydrate. A smaller glucose spike means a smaller insulin response. A smaller insulin response means less activation of the fat storage signaling cascade. Specifically, less lipoprotein lipase activation in adipose tissue and less inhibition of hormone-sensitive lipase, the enzyme responsible for releasing fat from storage. The Okinawan centenarians were not managing their insulin. They were choosing the carbohydrate that their food culture had always used, that happened to have a glycemic profile that never created the chronic insulin elevation that drives visceral fat accumulation. The biological consequence of 70 years of postprandial glucose curves that never spiked above the level that sweet potato produces is the near-zero visceral fat the researchers found. The anthocyanins, purple pigment polyphenols, in purple sweet potato add a second mechanism: documented anti-inflammatory and antioxidant activity that further suppresses the cytokine environment that drives fat storage. The carbohydrate was not just glycemically favorable, it was delivering bioactive anti-inflammatory compounds with every serving. Food 7. Fatty fish, salmon, sardines, mackerel, 3 times weekly minimum. Omega-3 fatty acids, EPA and DHA. A do not just support cardiovascular health. They are documented to maintain brown fat activity, the thermogenic capacity of adipose tissue that declines with age, and with the omega-3 deficiency that characterizes the Western dietary pattern. Research on EPA and DHA supplementation in older adults found that omega-3 status was directly associated with brown adipose tissue activity. The same compounds that reduce systemic inflammation and protect vascular function also preserve the thermogenic capacity that determines baseline metabolic rate. the rate at which the body burns calories at rest. The traditional Japanese consumption of fatty fish, not as a high-protein strategy, but as the primary animal protein of the food culture, consumed in smaller amounts across multiple meals, rather than in large portions once, delivered consistent EPA and DHA at the levels that maintain brown fat function. The centenarians who consumed small fatty fish several times weekly, as ordinary daily eating were maintaining thermogenic capacity that sedentary Western populations, deficient in omega-3s, progressively lose through middle age. The structural framework, Ichiju Sansai and Hara Hachi Bu. The 7 foods would produce a partial outcome in isolation. The Japanese eating framework that structures when and how they are consumed amplifies every mechanism. Ichiju Sansai, 1 soup, 3 sides, is the traditional meal structure that has governed Japanese eating for centuries. 1 bowl of miso soup. 1 primary protein, fish, tofu, natto. 2 small vegetable sides, typically including a fermented preparation and a cooked vegetable. Rice as the base. This structure naturally creates a plate that is high in fiber, moderate in protein, varied in bioactive compound delivery, and portion-controlled without any conscious measurement. The fiber arrives before the starch. The probiotic food arrives at every meal. The vegetables accompany every protein. The meal is consumed in a specific sequence that slows gastric emptying, moderates glucose absorption, and maximizes satiety signaling before the rice is reached. None of this is deliberate nutritional strategy in traditional Japanese culture. It is simply the meal. Hara Hachi Bu, the Okinawan practice of stopping at 80% fullness before every meal, adds the behavioral component. The brain requires approximately 20 minutes to register satiety signals from the gut. Stopping at 80%, when the awareness of fullness is just beginning to emerge, means that by the time the meal is fully processed, the sensation of having eaten enough arrives rather than the sensation of having eaten too much. Practiced at every meal across a lifetime, this creates a consistent 10 to 15% caloric reduction, without any experience of restriction. The combination of Ichiju Sansai and the Hara Hachi Bu produces something that no Western dietary protocol has replicated through explicit instruction. Portion control that emerges automatically from structure and cultural habit, rather than from willpower, calorie counting, or the discipline that cannot be sustained across decades. The longevity connection. Everything described above addresses fat accumulation. But the same mechanisms extend directly to longevity outcomes. The gut microbiome that natto, miso, and fermented pickles maintain is the same microbiome associated with lower all-cause mortality in centenarian populations. The butyrate produced by a well-fed gut microbiome is the same compound that maintains the intestinal barrier integrity, that prevents the low-grade endotoxemia, driving systemic inflammation across the lifespan. The UCP1 activation from EGCG and fucoxanthin is the same thermogenic mechanism associated with lower visceral fat, and lower visceral fat is the single metabolic variable most consistently associated with a longer healthy lifespan across every population studied. Hara Hachi Bu activates FOXO3, the longevity gene variant found at unusually high frequencies in the Okinawan centenarian population. Through the mild caloric restriction that stopping at 80% creates. The connection between caloric restriction and FOXO3 activation runs through AMPK and the mTOR suppression that extends cellular lifespan in every animal model ever tested. The centenarians of Okinawa were activating this pathway at every meal for 100 years without ever hearing of FOXO3 or AMPK or caloric restriction. Japan does not have a 4% obesity rate because the Japanese are disciplined. They are not. They have a 4% obesity rate because the food system they maintained, the specific foods, the specific preparation methods, the specific meal structure, the specific behavioral habits around eating, creates the metabolic conditions that prevent fat accumulation automatically, without effort, across a lifetime. And because the same system also prevents the chronic visceral inflammation that drives every major age-related disease, the country that never gains weight is also the country that lives longest. This was not designed, it was simply eaten. Which of these 7 foods are you already incorporating into your daily meals? And which one would you add first? Tell me in the comments below. Subscribe to Longevity Code. Every week we decode one longevity population, one food system, one set of mechanisms. Always peer-reviewed, always practical. No supplements to sell, no products to push, just the code.